In this video, we're going to talk about phloem. Phloem moves material from a source can be anywhere, to a sink, and the sink can be anywhere. In fact, the same phloem can be used in the opposite direction if the plant needs to move material in the opposite direction. Phloem cells are really pairs of cells. They're companion cells and sieve cells. The companion cells have a nucleus and lots of mitochondria and do most of the real biochemistry. Because amazingly, the sieve cell cytoplasm is the phloem sap that moves through the plant. These phloem sieve cells have what are called sieve plates, which are real large spaces in the cell wall to allow this phloem sap or phloem cytoplasm to travel freely through these continuous tubes of connected cells. This is a transmission electron micrograph of a companion cell and the sieve tube member. So here's the companion cell there, and here's the sieve tube member. Here's a picture of the sieve plates, and those are big gaping holes that do not impede the movement of phloem sap, and they're found. So there's a cross section of this space right here, this area right here, which is the cell wall that, to a certain extent separates these cells. Here's the 212 cartoon of this process. So there are locations within the plant that are, at any given time, source or sink. A source is either a set of cells that are making a lot of sugar or a set of cells that have stored quite a bit of sugar. They have to have enough sugar to actually load the phloem. Loading of the phloem with sugar creates pressure. How does it do that? It creates pressure because you're adding solute to the phloem, and water follows. Now, this is not a situation where I can make up problems that involve equilibrium, because this is a system that's not in equilibrium. You've got water coming into the phloem cells at the source, and then the sap, the solution that includes water and sugar and whatever else has been added in there, moves towards some other location in the plant. And that other location in the plant, if it starts to remove some of the sucrose and whatever else is being delivered, then that is a sink that is receiving the goodies through the phloem transport system. There's less pressure in the phloem at the sink than there is at the source, and that's why you get that sap moving. You notice that there have to be cells that can deliver the sucrose, but there also have to be cells that can take up that sucrose. Sucrose is one of many sugars it could be. But you have to be able to take it up and out of the interstitial fluid so it doesn't build up, and you have to be able to add it to the interstitial fluid so that there will be enough to generate the pressure at the source. This is a textbook version. Notice that it uses the expression active transport. That's not, and that the companion cell is gigantic compared to the actual phloem sieve plate cell, but the opposite is true. This is just a cartoon. And this active transport is kind of ambiguous. I mean, in which cell is ATP being spent? And here's the deal. If this is a cell, not all plants do this, but if this is a plant whose cells import material actively into the companion cell, that's the ones we're talking about. There are plants that actually it doesn't go into the interstitial fluid and then back out. They're actually connected together by something called plasmodesmata. But for 212 purposes, the active transport is ATP being spent by the companion cell to import lots of sugar. The sink cell, it has a root cell here, but that could just as easily be a baby leaf or any other growing tissue, right? Because any tissue that can't support itself has to be a sink, has to rely on the photosynthetic capacity or the photosynthetic storage capacity of some source. This is a cartoon of phloem loading. So this is happening at the This is where sugar is being taken from the interstitial fluid into the phloem cell. Protons are used, and so therefore you've got to have generated a proton gradient. Protons are used as the driving ion that allows sugar to move in against an electrochemical gradient. Now, sugar is a neutral molecule, so this is a concentration gradient against which sugar is moving. Water comes in because there's more sugar inside now than out. The fact that there's more sugar inside than out is a necessary component to the logic of this being a pressure generation system, right? If it was a channel, then the concentration gradient would have to be in the other direction. There would have to be more in the interstitial fluid than in the cell. If that were the case, then water would move in the other direction, since water, in this case, is the primary solute. So, water comes in, pressure builds. The other end, no ATP is required for this process. No energy needs to be spent to let sugar out. provided that sugar is getting used by cells at the sink, taken up by other cells at the sink. And when you release sugar through these channels, you release water. Water will follow because of the change in osmotic potential in the cell. It goes up, leads to water leaving, so pressure potential goes down.